this video to clarify some things we have talked about in class today, and it is dealing with DNA organization and how DNA is packaged and arranged inside of your cells. So if we were to look here at a strand of DNA, we would find its structure to be what is identified as a double helix or a twisted ladder. We would find that this would be held together by hydrogen bonds, and these hydrogen bonds will connect nucleotide bases from one end to another. And just to clarify, A's and T's get paired together, and C's and G's also get paired together. Now, where this gets tricky is that since this is a double helix, it wraps around itself. So if I were to take a strand of DNA here, it would continually wrap around itself to the point where it would begin to twist on itself. If you remember, we took two people, we took a string between us, and we both twisted in the clockwise direction, and this DNA began to twist up on itself. Well, what we see is that this DNA gets wrapped around these proteins called histones. These histones help twist and package the long strands of DNA in your body, so it will make it easier for us to replicate later on and divide. So, looking at one of your cells, we will see a nucleus, meaning that we are eukaryotic. And inside of your eukaryotic cells, you're going to see all of your DNA floating around. It's not really packaged nicely because it doesn't need to be right now. It's open, it's being read, it's making proteins. But as we start working towards replication, we're going to start to bundle it up. If you see this loosely floating around, we refer to this as chromatin. So this is where it is loosely floating around. Now once we start to package it, draw another cell here, another nucleus, we are going to find that they begin to look like rods, or what we refer to these as are chromosomes. Now you have 46 chromosomes, or 23 pairs. Now if I'm trying to double my DNA, I'm going to need to make twice as much. So what happens is your DNA begins to replicate, and you begin to see these X structures. So sometimes they are just drawn as X's, and this is where our vocabulary is really going to come into play as we take a closer look at one of these X's. You're going to find tightly bundled DNA, as if it is the shape of an L or two L's standing back to back. We call this entire structure a chromosome, and it is bound together by a centromere. The centromere acts as if it was a thumbtack, sticking them together so one strand does not get lost in the abundant space of your nucleus. So these two are connected. Now where this gets a little tricky is that you can think of these as two halves. So if I have my first half here, this is called a chromatid. The other half is also called a chromatid, but they are identical. So we replicated them. We've doubled the original strand, and you can think of these as twins, or, as you're going to see them referred to, sister chromatids. So these are our sister chromatids. They contain the same amount of DNA, but once I go to split my cell from 1 into 2, draw some nuclei here. Once I begin to split, I will pull these apart. So I'll draw my X here. And once I pull them apart, I will have the same amount that I started with, allowing me to have 2 identical cells at the end of cell division.